Alright, so welcome to the chapter 3 of our lecture in genetics. So today we're going to discuss a 3-unit or a 3-part lecture on this topic. Unit 1 involved the basic Mendelian genetics, wherein we're going to discuss the experimentation of Gregor Mendel that led him to develop or to establish the transmission genetics. We're also going to discuss some genetic crosses, monohybrid to trihybrid or polyhybrid crosses, alongside with back and test crosses, as well as the 3 laws of inheritance and the laws of probabilities. On Unit 2, we're going to discuss the autosomes and sex chromosomes. And lastly, for the Unit 3, we're going to discuss the non-Mendelian inheritance. So these days, scientists know how you inherit characteristics from your parents. They are also able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from your parents or even from your family history. This was of course made possible by Gregor Mendel. Gregor Mendel was the one who proposed the what they call transmission genetics, and he was able to perform this by breeding the pea plants or Pisum sativum he was growing in the monastery's garden. After all, he is an Augustinian monk. He discovered the principles that rule heredity, in which we know now as what we called Mendelian genetics. So Mendelian genetics is also known as transmission genetics, in which it is the principle of describing how traits are passed from parent to offspring. Of course, it was proposed by Gregor Mendel, as stated earlier, and Mendelian inheritance technically, or it refers to the certain patterns that are passed down from generations to generations. So who is exactly Gregor Mendel? So he is an Augustinian monk and a biologist, and because of his contributions on Mendelian genetics, he became the father of genetics. But a quick trivia, after publishing his work, it was not recognized until the turn of the 20th century, when a group of scientists reopened his proposed theory or his published work, and they began to realize that what was proposed by Mendel is the most plausible and the most acceptable in the scientific community. So Mendel's experimentation on transmission genetics began between 1856 and 1863, and between this span, he cultivated and tested around 28,000 garden pea plants. He also found out that the plant's offspring retained the traits of the parents, in which such traits of the parents are passed down from different generations. At the time of his experimentation, he was not aware of these particles, and he just called it particles that are inherited from one parent to another. And now, given the advancement in the scientific research, we know now that these particles he was referring to were actually chromosomes and your DNA. Mendel was able to establish these through different types of genetic crosses, and he was able to establish this by using a model organism known as Pisum sativum in scientific name, or peas by common name. So peas, he used this type of plants because they can be grown in a small area. He cultivated these peas in a very small space in his monasteries, and in a garden in his monastery, and these already produce a lot of offspring in a given period of time. So through this piece, he was also able to produce pure plants, which he allowed to self-pollinate through several generations, and these model organisms can be artificially crossbred. Just a quick botany in this one. The plant in this one, as you can see in this illustration, they contain pollens. No, pollen contains the sperm that is produced by the stamen, as you can see in this one. And of course, in a given plant, they also contain the ovary, which contains the egg. If the pollen pollinated technically the ovary, they can undergo what we call self-fertilization. And if the plant undergoes self-fertilization, they will be producing pure, or a plant or an organism containing pure strains. So I'm discussing a little bit of this background because Mendel involved these experimental methods wherein he hand pollinated the flowers using a paintbrush as depicted in this image. Particularly, he snipped or he cut the stamens to prevent self-pollination of this plant, for example, and covered each flower with a cloth bag in order to prevent contamination from the other pollens coming from the other plants and to ensure that he will be pollinating from his plants, from his observed plants. Through this, he was able to trace the traits through the several generations, and these traits that we're actually talking about are technically summarized in some of the genetics book. I will be showing that to you later on. So, how Mendel began? Mendel produced a pure strains by allowing the plants to self-pollinate for several generations. So, technically, these plants, if they self-pollinate, they will be producing a pure strain for several generations until such time that Mendel will be able to crossbreed other F2 to F3 generations. But anyways, Mendel began observing different pea plant traits to determine which characteristics were inherited and which characteristics were not. And these traits that I was referring to are actually in this table. These are some of the few examples we have from the seed shape all the way from the stem height. This column of F1 and F2 results are technically the offspring from the first and the second generations, and some of these detail are presented in a ratio. We call this a genotype ratio. You will be conducting some of these sooner or later. Mendel particularly used the term parent or P1 to F2 generations. When we say parent or P1 generation, this is actually the original plant that he bred. And do take note that parent generation contains pure strains. And then the resulting offspring of our F1 generation is none other than the F1 generations, as you can see. Most likely, when we cross two pure parent plants, one that is pure dominant or one that is containing a homozygous dominant and one containing a homozygous recessive rather, when we crossbreed these two parents, most likely the F1 generations are all heterozygous, contains heterozygous allele. So, with that being said, if they are heterozygous allele, they will be containing one dominant allele. So, that being said, what was reflected in all of these F1 generations are all dominant allele as well. So, F2 generations actually are organisms, or technically the generations resulting from breeding from two parents from F1 generation. And as we can see in F2 generations, there's a recessive trait that reflected or expressed in one of the offspring. So we've been talking about genetic crosses, and there are many different types of genetic crosses. We have from monohybrid to polyhybrid crosses, and this varies depending on the number of traits that we will be crossing. Mono, we will be crossing only one single trait from both of the parents. Dihybrid cross 2 traits, and polyhybrid cross 3 or more traits. So genetic crosses are usually carried out through a what we call Punnett square, which is just a diagram that is used to predict specific combinations that we can actually arrive to from given the genotype of a given parent. So in making a Punnett square, let's consider this problem. Determine the genotype and phenotype ratio of the resulting offspring if both of the parents contain heterozygous yellow pea allele. Before we start, of course, it's very important for you to familiarize yourself with some terminologies. When we say traits, technically, these are characteristics that of an organism. So these may refer to hair color, seed color, or shape, flower color, flower shape. All characteristics that are technically expressed in a given organism are known as traits. And in each given traits, do take note that they contain two alleles. So for this one, For this example is a trait for the color of the peas. Let's say this is a pea color trait, and then in each trait it contains two different alleles, each of which are represented by an uppercase letter or a lowercase letter, or two uppercase letters or two lowercase letters. It depends on the organisms. One of the traits may contain dominant allele, and when we say dominant allele, it is usually represented by an uppercase letters. These will carry the trait that is usually expressed in an organism. If there are two types of allele in a given trait, let's say like this one, the dominant allele will be expressed in an organism. For example, that this uppercase letter is a dominant allele which carries the yellow P allele, and this lowercase letter technically is the recessive allele which carries the green P allele. If there is a dominant, if there's a dominant allele in a given trait, it is the dominant allele that will be expressed in an organism. So with that being said, this plant will have a yellow peas, since the yellow pea is the dominant allele over the recessive one, which is the green allele. The recessive allele, on the other hand, is represented by lowercase letters, and this carries the trait that does not usually appear in an organism. It does not produce a trait at all when only one copy in a given trait is present, especially when it is with a dominant allele. For a recessive trait to be expressed in an organism, two copies of this recessive allele must be expressed in a given trait. In this example, the recessive allele carries the green pea trait, but since, as I said earlier, we have here a dominant P allele which carries the yellow P allele, the plant will express yellow color in its P. Lastly, some of the last reminders here, you might encounter terminology such as heterozygous, homozygous dominant, and homozygous recessive. When we say heterozygous, the plant or the parent carries two different types of allele, one that is dominant and one that is recessive. When we say homozygous dominant, the plant may carry two dominant or two uppercase letters, as you can see in this one. And when we say homozygous recessive, the plant carries two recessive allele, as you can see in this one. So this is the only time that the plant will express the recessive traits. So going back to our step-by-step process in making a Punnett square, considering this problem, as we can see, step 1 is to make the grid or make a 2x2 Punnett square just like this one. So place the alleles of the gametes of one parent along the top portion of the grid and those of the other parent along the left-hand side, as you can see in this one. For your visualization, I particularly created this one wherein the parent one carries heterozygous yellow P allele. Since both of the parent carries heterozygous yellow P allele, so meaning to say one of which is dominant, which is the yellow P allele, and one of which is recessive, which is the green P allele. So in making your Punnett square, just input both of these allele into our table, as you can see in this one. One is in the column and one is a row. Step 2 is basically to fill in the grid. Just combine the parent alleles from both of these grids and you will be able to come up with a genotype and phenotype ratio. But for the purpose of this lecture, I particularly assign numbers and letters in each table, as you can see. By means of combining the alleles, the first step is technically to take the first allele of the first parent, or the y1, and multiply it to the first allele of the second parent, which is technically the y3, and our answers are reflected in box A in this one, which is technically y. This one is homozygous dominant. The second step is just to carry on with the remaining boxes. So we have here y1 multiplied with y4. Now we have dominant y and recessive y for our box C. And just carry on with the rest of the boxes. For step number 3, it's time for you to fill in the offspring by using the law of dominance. As stated earlier, you have to determine the genotype and phenotype ratio. This particular offspring will have a combination of a homozygous dominant, YY. With that being said, we are expecting that this organism will have the yellow P allele because it contains both of the dominant traits. On the other hand, two of the offspring might actually display a heterozygous allele. So with that being said, they contain one dominant and one recessive. And according to our discussion earlier, by using the law of dominance, if there is one dominant allele in our traits, the dominant trait will be expressed. Hence, the color of the P will still be color yellow. Lastly, this organism or this offspring inherited both of the recessive alleles of both of the parents. So with that being said, what will be expressed in the organism is the recessive trait, which is technically the green P allele. The next step is to determine the genotype ratio. This is just a very simple task because you technically have to segregate these different output. The genotype ratio is 1:2:1. The 1 here represents this one. Technically, we have 1 homozygous dominant. The 2 here represents 2 heterozygous. And the 1 here represents this one, which leaves us with only 1 homozygous recessive. The phenotype ratio is much easier than the genotype ratio because technically you will have to determine their appearances. How many plants will have the yellow P allele and how many plants will have the green P allele. Since 3 of them contains the dominant traits, as stated in our genotype ratio, 1, 2, 3, 3 of them contains the dominant allele. 3 of them will have yellow P allele. And only 1 contains the recessive traits or the green P allele. I particularly attach here a YouTube practice. You might want to click on this icon if you need more practice on setting up your Punnett square. These are some of the summary of terminologies that I used in the previous discussion, but I already explained them. So I just want to skip this part, but you might want to review this terminologies as it will help you to further understand our discussion. In this particular part of this recorded lecture, please stop this lecture and try to answer this by yourself. Alright, so the answer for this short exercise is technically 2:2 for our genotype ratio, and for our phenotype, technically we don't have a phenotype ratio because all of the offspring will have the same characteristics. Our problem here is that we need to predict our offspring if our parent 1 contains the homozygous dominant yellow P allele and the other contains the heterozygous yellow P allele. So parent 1 contains YY or contains both dominant allele, and parent 2 contains 1 dominant and 1 recessive. So we just have to put that in our Punnett square. This one technically represents parent 2. As we can see, 1 dominant and 1 recessive. This one technically represents our parent 1, which contains both dominant allele. So conducting or multiplying this one or conducting genetic crosses in between of our traits, we arrive to a genotype ratio of 2:2. So 2 here represents that there will be 2 homozygous dominant, and 2 here represents that there will be 2 heterozygous allele in our offspring. If we're going to predict which traits will be expressed in these organisms, since all of them technically contains at least one dominant allele. The dominant allele is technically the yellow piece. It means that our phenotype ratio, all of our offspring, as expected, will contain the yellow seeds or yellow P rather. Going back to the experiments of Mendel, he particularly crossed or he particularly observed the shape or the texture rather of the bees, as you can see in this one. um, Mendel wanted to determine the genotype and phenotype ratio of the F2 generation, which is technically the third generation from this cross. If both of the parents have homozygous dominant round seed allele, meaning to say parent 1 contains both dominant allele, having the dominant trait is the round seed, and parent 2 contains homozygous recessive, which is technically a wrinkled seed allele. So, what Mendel conducted was to cross these two parents, and if we're going to predict the offspring or the F1 generation from this through a Punnett square, the resulting offspring will be like this one. So, if we're going to cross two pure parents or two homozygous dominant and homozygous recessive, all of the offspring will contain heterozygous alleles, which is already expected because we have here dominant and we have here recessive traits. And as such, 100% of our offspring will contain heterozygous allele. And if I'm going to ask you, what do you think is the characteristics of the resulting, or what do you think is the phenotype ratio of, or phenotype rather, of the resulting offspring from this cross or from this breed? You might want to pause the video if you want to answer. But the answer in this one is that technically all plants will contain the round seeds. Since all of them, round here or the round piece technically is the dominant traits, and since all of the offspring contains at least one dominant allele, they will be expressing round peas than the wrinkled seeds. Again, recessive traits could only be expressed if a specific traits contain two recessive alleles. On the other hand, Mendel is not yet finished on this one, as he wanted to know what will be the resulting offspring if he will be cross-breeding two offspring or two parents from the F1 generation. So RR technically times RR, and if we want to predict that one, we have to put that in our Punnett square just like this one, and this will be our resulting offspring, which is technically 1:2:1, or 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So our expected offspring would definitely look like this. So 3 of them, which is technically this one, and this one, would contain round seeds. Round seeds because each of them contains at least one of the dominant alleles, and only one of the offspring exhibited the wrinkled seeds. So some of these peas have a smooth texture while others are wrinkled because technically in the F2 generations, the recessive traits are already reflected. Now, let's talk about the test cross. So what is a test cross? So test cross is technically a continuation of what Mendel conducted. So last time, in the previous slide, we stopped from this generation, but in test cross, it further genetic crosses or further breeding of the plants from the F2 generation will be conducted. So there are two possible test crosses here. One that is between a homozygous dominant and a hybrid, or a plant containing the heterozygous allele, and one between a heterozygous recessive and a hybrid. The result, of course, if we want to predict that one, we will be using another Punnett squares in this one, and our genotype ratio is technically 1:1, in which we will be having the ratio of the homozygous dominant and the ratio of the heterozygous or hybrid is just the same. So this is 50-50%. The same can be observed in heterozygous recessive and homozygous recessive. I'm sorry, this one is a mistake. I would be editing this one. The same can be observed in homozygous recessive and a cross between a heterozygous, as you can see in this one. So, most geneticists nowadays, they utilize this test cross in order for them to determine a specific phenotype or genotype ratio of a given parent, especially if that parent has a known genotype ratio. So, for example, I have here a problem set. Determine the genotype of this plant with dominant yellow P allele. So, what is known technically is that this plant contains a dominant yellow P allele, but we are not sure if if this plant carries a homozygous dominant or a heterozygous allele. So if you want to identify this one, geneticists usually use test crossed because the resulting offspring will tell you everything you need to know about the genotype of that parent. So the first step in test crossing is that you breed the plant, or technically you breed the plant or the plant with a known genotype ratio with another plant. However, um, to an organism, you breed this plant to an organism with a recessive phenotype. So technically that is a small yy, as you can see in this one. And then if the resulting offspring are all exhibited the dominant allele, this means that our parent with a known genotype contains a homozygous dominant allele, this one, YY. On the other hand, if the offspring are technically 50-50, meaning to say 50% of them exhibited yellow P seeds or yellow P allele and 50% of them exhibited green seed allele, this means that the parent with a known genotype contains a heterozygous allele. So this could be predicted through the Punnett squares as well. Now, we're done with the test cross. In this part, we're going to discuss the back cross naman. Back cross technically is a cross between the F1 generation and the parent. In the test cross, this is a cross between two parents from the F3 generations. Now, if the test cross is used technically to determine the unknown genotype ratio of a given organism, back cross is used to usually back cross is being utilized by farmers as it can be helpful to them for the plant breeders to design the breeding as per the demand, or technically if they want to produce a specific strain of the plant according to the demand of the economy or according to the demand of the market. So how is backcross being conducted? So number one is to take one parent from one parent which is a pure which contains a pure strain, and take one organism from the F1 generation and conduct genetic crosses. And the resulting offspring, it would give you 50% pure or homozygous. It's either homozygous dominant if you take this parent, or homozygous recessive if you take this parent. And the other half would give you 50% heterozygous. So in summary of the results of monohybrid crosses, inheritable factors or genes are technically responsible for all heritable characteristics. Phenotype is based on genotype, but the differences is that in phenotype, technically it's the appearance of our organisms. Each trait is based on two genes, one from the mother and other from the father. And true breeding individuals are homozygous, in which both alleles are either dominant or recessive and are pure. Now, what are the three laws of inheritance? These three laws of inheritance are very important to consider, especially here in Mendelian Genetics, because this one technically holds the keys for some explanations regarding inheriting characteristics from parent to offspring. The law number one is the law of dominance. In this one, in a cross of parents that are pure for contrasting traits, only one form of the trait will appear in the next generation, and all of the offspring will be heterozygous and express only the dominant trait. In summary, law of dominant states that the dominant traits will always be expressed in an organism. As you can see in this one, all of our offspring from the F1 generations exhibited the dominant trait because technically a cross between a homozygous dominant and homozygous recessive will give one dominant allele to each of our offspring. So with that being said, all of the dominant alleles here will be expressed. The law of segregation, on the other hand, states that during the formation of gametes, the two alleles responsible for a trait separate from each other. So for example, this is from your mother and this one is from your father. Technically, this one separates, the two traits technically separates, and they are the ones that are being randomized during genetic process. This one is basically your Punnett square. So alleles for a trait are then recombined at fertilization, producing the genotype for the traits of the offspring. So that's a very random possible combinations. In law of independent assortment, to summarize, it states that the traits are inherited independently and these traits are never in group. So for example, if your mom has curly hair and your dad has freckles, and of course, the resulting offspring could be a boy with curly hair and freckles. So technically, these traits are inherited independently and they do not come in group. So the law of independent assortment can be illustrated further using what we call dihybrid crosses. In dihybrid cross, this is actually a breeding experiment that tracks the inheritance of two traits. In the previous example, we have the monohybrid cross, wherein we are trying to predict the possible gamete combinations the possible traits of a given offspring, considering only one traits. In this one, in the hybrid cross, we are now considering already two traits. So it could be either wrinkled seeds and yellow color, or yellow pea rather, or it could be color of flower color or flower of the seeds. It depends on the traits, but in the hybrid, again, we're talking about two traits. So this could be the dihybrid cross is a perfect example rather of law of independent assortment, wherein each pair of alleles segregates independently during gamete formation. So in conducting a dihybrid cross, we usually conduct a calculation wherein this is the actually formula 2 raised to n, where n is the number of heterozygotes, in order for us to predict the possible number of gamete combination in a given traits. So let's have an example. So how many gametes will be produced for the following allele arrangement? So do remember our formula, 2 raised to n, where n is the number of heterozygotes. You might want to stop this video and try to answer this on your own. Alright, so the answer here is, for the first one, it would be 4 gametes because there are 2 heterozygotes here. Substituting that to our formula, 2 raised to 2 is technically 4. In this one, I have here 1, 2, 3. We have 3 heterozygotes. The 2 double C is not heterozygote, that's actually a homozygote. So because it contains 2 dominant alleles, remember that heterozygotes are contains 2 alleles that are opposite to one another. So that's 2 raised to 3. So that's why the answer is 8 gametes. So 2 times 2 times 2. And in the last one, we have a total of 64 gametes. Okay, now in dihybrid cross, we also create a Punnett square in that one in order for us to predict the possible genotype and phenotype ratio of our parent. Let's consider this problem. Determine the genotype and phenotype ratio of the resulting offspring if both parents contain heterozygous yellow round P allele. So as we can see in our plant organism, the yellow and round P are reflected or expressed in our plants. This means that the yellow and round traits are both dominant traits or dominant alleles. Alright? In this case, we are already dealing with a hybrid crosses because we are now dealing with one, color of the P, and second, shape of the P. So this is already two traits. Now the question is asking us what could be the possible um, characteristics of these two combinations. So before we start, uh, again, in the hybrid crosses, each parent now carries two traits, and each trait now has four alleles. A while ago, in the monohybrid crosses, each trait carries two alleles. One could be dominant, one could be recessive. In the hybrid crosses, since we are now considering two traits, we will also be considering four different types of alleles. So in this example, the two traits contains heterozygous alleles, as you can see in this one, with each trait having one dominant and one recessive alleles. Alright? Next slide. So the trait RR, which is technically the trait 1, carries the shape of the seed, having round seed as a dominant trait, while wrinkled seeds as a recessive trait. These characteristics are just brief examples. Trait 2, YY, on the other hand, carries the color of the seeds, having yellow seeds as a dominant trait, while green seeds as the recessive trait. Now, to visualize our problem, I specifically created this representation, as you can see right now. Again, step 1 is to predict the possible gamete combination on each parent. So we're going to use our example 2 raised to n. So in this one, the number of heterozygotes are 2 because there are two heterozygotes in our parent 1, also 2 heterozygotes in our parent 2. Calculating these possibilities, we have 4 possible gamete combinations in each parent. So remember, there are 2 traits in this example. Trait 1 carries the shape of the P, while trait 2 carries the color of the P. Now, we have to predict the possible gamete combination on each parent. The first one is you have to employ the FOIL method. Step A is to multiply the first allele of trait 1 to the first allele of trait 2. Thus, our first possible gamete combination is RY, this one. Step 2, on the other hand, technically, is multiply the first allele of as you can see here, multiply the first allele of trait 1 to the second allele of trait 2. Thus, our second possible gamete combination is R, dominant R, and recessive Y. In step C, you'll have to multiply the second allele of trait 1, the recessive R, to the first allele of trait 2. Thus, our third possible gamete combination is R, recessive R, and dominant Y. In step D, you have to multiply the second allele, which is technically the recessive R of trait 1, to the second allele of trait 2. So this is recessive to recessive. Thus, our fourth possible gamete combination are recessive R and Y. So in this case, as you can see, just like when you were in elementary, this is technically a FOIL method, no? So you will have to multiply each of these traits together in order for you just to predict possible gamete combinations. So with that being said, since both of these parents are just the same, they contain the same genetic traits, both of these parents has these possible gamete combinations. This is based from our FOIL method. Now you'll have to take these possible gamete combinations and you will be putting them in our Punnett squares, just like the monohybrid cross. You will have to input both of these possible gamete combinations on the top and left corner. For the top corner, it's for either of the parent 1 or 2, And for the left corner here, as you can see, you'll also have to input these gamete combinations of the other parent, as you can see in that one. Okay, so just like the monohybrid cross, you can interchange, but in this point, it's pointless because technically they have the same gamete combinations. Alright, so fill in the grid and combine the parent alleles inside the boxes. So just like the monohybrid cross, we're going to take this one and then multiply it with this one. The resulting answer is this one, dominant R and dominant Y. Likewise, continue while you fill in the boxes. We're going to take this one and multiply this one to this one, and the answer will be this one. We're going to take this one and multiply it to the third column, and then the answer is this one. We're going to take this one and multiply it to the fourth column, and the answer is this one. So technically, you'll just have to follow the steps in each of the row of our Punnett squares. And if you notice, our Punnett square is now a 4x4 Punnett square. Now, you'll have to identify the genotype and phenotype ratio such that you will have to quantify each of these traits that are unique to one another. Like for example, this one represents the RRYY because there are only one dominant RR and dominant YY across our results in this one. And continue your way up to the second column, up to the third column, and fourth column until such time that you are able to account every single possible genotypic ratio. On the other hand, we also have here's the phenotype ratio, wherein you will have to predict the color and the shape of the seeds, or the pea rather. Again, if there is a presence of a dominant allele, it is the dominant allele that will be reflected. Like for example, in this one, this is dominant R and dominant Y. With that being said, this offspring or this possible gamete combination will have a phenotype of round and yellow, as represented by this one. On the other hand, in this example, we have recessive R and dominant Y. So that being said, this one will have a phenotype of wrinkled and yellow. So once that you're able to identify the phenotype of every single genotypic ratio of every offspring, you will have to quantify them as well and present them in a ratio. Like for example, in this Punnett square, there are 9 round yellow in this example. There are 3 round greens. There are 3 wrinkled yellow and only 1 wrinkled green. Thus, our phenotype ratio would be 9:3:3:1. If you find this a bit confusing, I particularly link a YouTube link on I particularly link a YouTube a short video on setting up a dihybrid cross in a Punnett square. Now let's move on to the trihybrid and polyhybrid cross. Considering this problem, determine the genotype and phenotype ratio of the F1 generation if both of the parents contain heterozygous yellow round seed and white flower allele, and parent 2 is homozygous to the recessive traits. Meaning to say that the parent 1 carries all of the dominant traits. The dominant traits with regards to color of the seed are yellow, round with regards to the shape, and white with regards to the color of the flower. So these are the dominant traits because they were expressed in the organisms. On the other hand, the phenotype of the parent 2 is the exact opposite of the phenotype of parent 1 because according to our problem, they carry all the recessive traits. Now before we start, in trihybrid and polyhybrid crosses, each parent carries multiple traits and multiple alleles. Since this is a trihybrid, this parent now carries 3 traits. Trait 1, which is represented by the letter A, carries the traits for the seed shape. Trait 2, as represented by letter B, carries the traits for seed color. While trait 3, as represented by letter C, carries the traits for flower color. The more traits that we consider in a given cross, the more possible number of gamete combinations. And the more possible number of gamete combinations, the larger our Punnett square will be. For the monohybrid cross, we only have a 2x2 Punnett square. In the hybrid, it's 4x4. In this one, it's 8x8 Punnett square. But before we jump into creating our Punnett square, it's important for us to predict the possible gamete combinations. Applying our formula, 2 raised to n, wherein n here technically is the number of heterozygotes, and since there are 3 heterozygotes in this example, 2 raised to 3 is equal to 8. So meaning to say, we need to predict at least 8 possible gamete combinations for this problem in order for us to set up our Punnett square. So in predicting possible gamete combinations like this one, you'll have to take letter per letter in each traits. Step A, take the first letter of the first trait and multiply it to the first letter of the second and third trait. In this example, we got all the dominant traits of letter A, B, and C. Step 2 is take the first letter of the first trait and multiply it to the first letter of the second trait, as well as to the second letter of the third trait. And as such, we got dominant traits A and B, but the recessive letter C, as stated in this one. Step C, take the first letter of the first trait and multiply it to the second letter of the second trait, as well as to the first letter of the third trait. Now, in this example, we got the dominant A and C, but the recessive B, which brings us to our third possible gamete combinations. In step D, you will have to still take the first letter of the first trait and multiply it to the second letter of the second, as well as to the third trait. In this one, we got only one dominant traits, which is letter A, and the rest of the traits contains recessive traits B and C. In step letter E, since we already have 4 possible gamete combinations, but according to our calculations, we need 8, so we still continue. In step E, you take the second letter of the first trait and multiply it to the first letter of the second trait, as well as to the first letter of the third trait. In here, we got one recessive letter A, and the rest of our traits are dominant. Step F, take the second letter of the first trait and multiply it to the first letter of the second trait, as well as to the second letter of the third trait. Our sixth gamete combination contains 2 recessive letter A and C, but a dominant letter B, as we can see in this one. For step G, take the second letter of the first trait and multiply to the second letter of the second trait, as well as to the first letter of the third trait. In this possible gamete combination, we have 2 recessive traits, recessive A and B, but dominant letter C, as seen in this one. Our last possible gamete combination, take the second letter of the first trait and multiply it to the second letter of the second trait, as well as to the second letter of the third trait. In this case, our possible gamete combinations are all recessive letter A, B, and C. Once that we are able to predict the possible gamete combinations of each parent, we can now be able to create our Punnett squares. So these technically are the 8 possible gamete combinations that we calculated earlier from parent 1. And since parent 2 only contains a recessive traits, as you can see, if you apply the same method, you will be arriving in the same answers. It's all recessive traits. And as such, just continue creating your Punnett square. For example, if I'm going to take this one, the cell number 1 of this column, I'm going to multiply it to cell number 1 of this row. The answer between these two is this one. And continue moving through each boxes until such time that you will be able to finish and fill in the possible gamete combinations. Probability and genetics. So Mendel was the first person to realize that probability can be used to predict the results of genetic crosses. In other words, he could use probability to know that all of the offspring of the first generation would be tall without even seeing them and just mere calculations. Now we have two we have two portions, or technically two rules in probabilities. It's either rule of multiplication or rule of addition. In rule of multiplication, technically we use this for independent events in sequence. No? So let's have an example. In a cross between pea plants that are heterozygous for purple flower color, what is the probability that the offspring will be homozygous recessive? So we are being asked to calculate the probability given the traits of these parents. So let's try to understand the following traits. So what was stated in this one is that if we have these parents, what is the probability that their offspring will be homozygous recessive? So to be honest, a Punnett square can actually tell you the answer. So let's try to build a Punnett square here. So I have here 4 boxes. This one is blank. And then our Punnett square. Letter P, recessive. And then begin constructing your Punnett square. Alright. The question here is we are only interested with the homozygous recessive. But if we're going to analyze our Punnett square here, to be honest, there are 25% of the offspring is homozygous dominant. On the other hand, 50% could be heterozygous. And lastly, there is only 25% as well by percentage that our offspring will be homozygous recessive. So the answer in this one is 25%. 25%. Or technically 1/4. Or 1/4. Either way is okay. Now, this one may take some time, especially for answering an examination. There's another method to follow, which I call this cheat sheet, where I follow technically this derived answers from every possible parent crossbreeds. So for example, if you cross a heterozygous and a heterozygous parent, 25% or 1/4, or there is a 1/4 or 25% that the offspring will be homozygous recessive, and 25% would be homozygous dominant, and 50% would be heterozygous. So technically, this one, the ones that we created, are the same with this one. However, if you cross two homozygous parents, let's say pure dominant multiplied with pure recessive, you will be arriving to 100% heterozygous offspring. On the other hand, we have pure dominant and pure dominant. Of course, our offspring will be 100% homozygous dominant. But if it's pure recessive and pure recessive, our offspring will also be 100% homozygous recessive. However, in the cross between heterozygous and homozygous, in this one, 50% or 1/2 of the time, or 50%, there is a 50% chances or probability that the offspring would be homozygous dominant, and the other 50% are technically heterozygous, right? So if we're going to apply the cheat sheet, this one, in our question, so it's PP times dominant P, recessive P. What are the odds that the offspring will be homozygous recessive? You just have to look in our cheat sheet. You can actually memorize this one. So since they are both heterozygous parents, these are both heterozygous parents, we're going to look at this portion, right? Because it's heterozygous times heterozygous. And according to our cheat sheets, there will be 25% that the offspring would be homozygous recessive. And that's your answer, right? It's easier this way. I think you just have to memorize this. Let's have another example, especially if it's already a dihybrid cross, right? So this one is an example of a dihybrid cross. in a cross between pea plants that are heterozygous. So both of our parents are heterozygous as well for two traits, purple spotted flower. What is the probability that the offspring will be homozygous recessive? So technically, if I'm going to write that down, that's Y, R, R. Again, as discussed earlier, this could be, sorry, let me just complete the equation. Let's say, as discussed earlier, maybe this specific trait is trait 1. This hold the trait for the color of the flower. And this trait technically is trait 2, which holds the trait for the spots or texture of the flower. So the question is asking, what are the odds that these two parents will be producing an offspring containing homozygous recessive, right? Now, I'm not going to conduct, you can actually create a Punnett square and you will be arriving in the same answer. But supposedly that this is an examination, you no longer have time to create a Punnett square. And that is the time where our cheat sheet comes into play. First, determine the parents. Parents are technically homozygous, right? So this is a crossbreed between homozygous and homozygous. The technique in dihybrid crosses is that first you'll have to determine or calculate them trait by trait. And it asks, what are the odds that it will be producing recessive traits? Let's first determine trait 1. So YY, or the heterozygous Y, multiplied with the heterozygous Y, what are the odds that we will be arriving to our recessive traits? Let's look at our cheat sheet. So we should be looking on this data because both of our parents are heterozygous. And then we are looking for a homozygous, for the probability of homozygous recessive, which is technically this one, right? So, with that being said, for this alone, we have 1/4 or 25% chances. Now, we still have trait 2. So, trait 2 here is technically this one multiplied with this one. What is the chance that they will be having this? Look at our cheat sheet again. So let me just delete this one. Cheat sheet again. So both of our parents in R are technically heterozygous. So we're going to look at this data. And we are looking for a homozygous recessive, which is also this one. So it's also 1/4. So 1/4. Now we have two data already. One from trait 1 and the other from trait 2. Now, what you can do here is technically to apply the rule of multiplication. So 1/4 times 1/4 is 1/16. Now, in most genetic books, it is actually preferred to have this or express this in percentage. So how do you express this in percentage? Simply divide 1 by 16 and then multiply it by 100. The answer is 6.25% or 1/16. Alright. Let's have another example for trihybrid crosses. So in a cross between BBC and the other parent, what is the probability that the offspring will be this, which is technically an offspring which contains heterozygous alleles. So again, I'm not going to conduct my Punnett squares here, but you can actually try to construct a Punnett square and you will arrive in the same answer. But in this one, for trihybrid cross, I'm not going to construct any Punnett squares since it's too time consuming, especially if this is an examination. So according to this one, the answer is 1/8 or 12.5% chances. But let's try to calculate that using our cheat sheet. So, I have here AA. Let me just copy my equation. AA, BB. Do I got that correct? Pure heterozygous. Multiply AA, BB, and CC. what are the odds that they will be producing the same trait as the parent one, which is technically heterozygous allele to all traits. Alright, let's apply our technique. So let's first calculate them trait per trait. So we have 3 traits here. Trait number 1, multiply with this one. Trait number 1. What are the odds that they will be producing this one, which is technically heterozygous as well? Let's look at our cheat sheet. Heterozygous times heterozygous, which is this one. And we what are the odds that they will be producing another heterozygous, which is technically this one? So the answer is 1/2. So I'm just gonna input here 1/2 plus. Let's move on to the second trait. We have here trait number 2 multiply to this one. Now this one is a different combination because we have here one heterozygous and one homozygous dominant. So let's try to look at on our cheat sheet, which is this one, since one heterozygous and one homozygous dominant. So we are currently looking for the probability of this one. If we cross this one to this one, what is the probability that they will be producing this, which is a heterozygous? And since we are currently looking at this one and we want heterozygous, the answer is 1/2. So plus 1/2. Now we are done with our last trait, which is this one and this one. This is a cross between a heterozygous and a homozygous dominant. The same with the previous one. If we're going to cross this one and this one, what are the odds that they will be producing this? Right? A heterozygous as well. So we're going to look at this cheat sheet because it's heterozygous times homozygous. And we are currently looking for this one, for the heterozygous. So the answer is 1/2 plus 1/2. Oh, I mean multiply because this is rule of multiplication. So 2 times 2 is 4 times 2 is 8. The answer is 1/8. The same answer with this one, right? So 1/8, you can actually convert that to percentage. 1 divided by 8. The answer should be multiplied by 100. The answer is 0.125. We multiply it by 100. The answer is 12.5%. So meaning to say there are 12.5% chances that the offspring will be the same with the parent one. Okay. In rule of addition, essentially they are just similar with the rule of multiplication, but from the name itself, of course, we use addition. So we will also be using our cheat sheets here, but just another definition, rule of addition is usually applicable for mutually exclusive events, or meaning to say 2 or more events that cannot happen simultaneously. Let's have an example. In a cross between pea plants that are heterozygous for purple flower color, what is the probability of the offspring being heterozygote? Converting that into an equation form, we have heterozygous alleles. Both of them are heterozygous alleles. Both parents are. And then it is asking what is the probability. What is the probability that our offspring will be heterozygote as well, right? So you can create a Punnett square, and to be honest, I already know the answer from our past calculation, and I do hope you also know the answer with this one. So I have here P homozygous dominant, heterozygous, homozygous recessive. So the question is asking about this heterozygous recessive, which is technically this. By percentage, it is 50%. So the answer here is 50% or 1/2. Next, in a cross between these traits, what is the probability that the offspring will be AA will be the following? I don't want to read that anymore. So I'm going to try and copy this to another page, which is this one. So we will have enough space to calculate. Okay, so the offspring that we wanted to find is either the other one or the other one, which is this one or this one. Okay, so I have to write it pala talaga. AA, PB, CC, technically heterozygous times AA, PB dominant C. What are the odds that the offspring would be this or this? Okay. Now, in rule of addition, when you see the word or, normally you will be using the rule of addition than the rule of multiplication. But if you see the word and, you will be using the rule of multiplication. So as you can see, based from our previous definition of rule of addition, mutually exclusive events, meaning to say events that cannot happen at the same time. So technically, they are actually describing this one. These are events that cannot happen at the same time, because it's asking you what is the probability that you will have either this one or this one. But anyways, going back, let's calculate the probability of these mutually exclusive events using our cheat sheets once again. So let me just transfer this one so I could access my cheat sheets easier. Just like the rule of multiplication, we're going to calculate this by traits per traits. So starting with letter A, and then cross that one with the other parent, which is technically this one. This is a cross between heterozygous parents. And we wanted to know what is the probability if we cross two heterozygous parents, what is the probability of the offspring to have a homozygous dominant trait. So going back to this one, both of our parents is heterozygous, and we wanted to know if what's the probability of our offspring having homozygous dominant traits. Technically, the answer is 1/4. This one is... Oh, wait, let me... Oh, no. Let me just delete that. Okay. So technically, the answer in this one is 1/4. Now, we're not yet done because we still have two remaining traits left. I'm going to calculate this one as well as with this one. As you can see. So this is a cross between heterozygous parent and a homozygous parent, as you can see here. And what are the odds that they will be producing this, which is heterozygous? Now let's try to look that up in our cheat sheet. So this is a cross between heterozygous and homozygous. So hetero times homo, which is technically this one. And what are the odds that the offspring would be a heterozygous? The answer is 50% or 1/2. So I'm just gonna write it down in this one, 1/2, right? Last trait I have here, this one, which is heterozygous, multiply with a homozygous. Technically the same, but let's not skip the process. So hetero times homo, technically this one. And you wanted to know what are the odds between these two parents that will be producing a heterozygous offspring. So technically the same answer. So that's 1/2 as well. 1/2. Now set that aside. Now we need to calculate as well this one, the other possibilities. So but let's set that aside, our information so far. Oh, sige, let me delete na lang so that 1/2. Okay, 1/2. And then I still have this other side that needs to be calculated. So I have here AA, BB, CC multiply with AA, BB, and CC. Working our way towards these traits, first starting with letter A, multiply with this one. So what are the odds that they will be producing these traits? Hetero times hetero, and we're looking for homozygous dominant. Going back to our cheat sheets, technically hetero times hetero, and we're looking for dominant. The answer is 1/4. 1/4. And then moving on to the next trait, we have letter B here, which is hetero times dominant. What are the odds that they will also be producing a dominant trait? So hetero times homozygous dominant. So we're looking here on our cheat sheet. What are the odds that they will be producing a homozygous dominant? The answer is 1/2. 1/2. Lastly, I would be taking out the last traits, multiply this one. What are the odds that they will also have this homozygous dominant trait? So hetero times homo in our cheat sheets, that is this one, hetero times homozygous. What are the odds that they will be producing this, a homozygous dominant, which is this one? Technically, the answer is also 1/2. 1/2. Oh. Now, before we add, you add these two possibilities, but before you add them, you have to multiply each number in the parentheses. So let me just erase this one for more space and rewrite them. Oh no. So anyways, I'm just going to calculate. So anyways, 1/4 times 1/2 times 1/2 plus 1/4 times 1/2 times 1/2. The answer is this one, as you can see. So 4 times 2, that would be 8 times 2, that would be 16. So this one is 1/16. We have the same numbers here, so this one is 1/16 as well. So you'll have to add this one. All right, so going back, so for example, 1/16 plus 1/16 equals 2/16. And we're going to simplify that. The simplified form of 2/16 is 1/8. The answer is, of course, 1/8. So meaning to say, in the cross between these two parents, the probability of them that the offspring will be this one or this one would be 1/8. You can actually convert them into a percentage. So 1 divided by 8 times 100, the answer is 12.5%. Right? Okay, so when do we use the rule of multiplication? So when we have independent events in sequence, you multiply the probabilities of the independent sequential events. So for example, in here, in heterozygous pea plants, what is the probability that the offspring will be homozygous recessive? As we can see in this one, we only have independent events in this one that are separated to one another. But my clue, as I said before, if you see in the question the word "and", most likely you will have to use the rule of multiplication. But for example, what is the probability that the offspring form a cross between ABC, while parent 2 is WYZ, will be ABC and ABC? So for example, just watch out for the word "and". On the other hand, for the rule of addition, you will be using them for 2 or more mutually exclusive events. The clue word here is the word "or". So if you see the word "or" in your question, most likely you will have to use the rule of addition. Right? Oh, sorry. Okay. I have some another examples here. This one is much more difficult. not really that difficult. The question is, suppose that parent 1 carries heterozygous green stem, yellow seed, and white flower trait. So this is trihybrid cross. While parent 2 carries the homozygous recessive traits. What is the probability that the offspring will also contain the homozygous recessive traits? Now, again, as I said before, we can definitely carry that out through a Punnett square. But you can definitely, but you no longer have the time to create this very complicated Punnett square, especially if you are in the middle of an examination. You can just carry out on our cheat sheets, which is my derived formula that I created for you. So through cheat sheets, we are able to calculate it that the answer would be 1/8. And it's actually similar with our Punnett square here. As you can see, the Punnett square tells us that 8 out of 64 possible gametes combination will have the homozygous recessive trait. So 8/64 is technically 1/8. Essentially, if you're going to use the cheat sheet, it will also give you 1/8 answer. Essentially, the same answer with our Punnett square. So 1/8 is converted to a percentage, so that's equivalent is 12.5%. Okay, moving on from Unit 1, now we're going to discuss about non-Mendelian inheritance. So not all genetic offspring or genetic variations rather could be explained by Mendel's law. And there are some few exceptions that happens during the fertilization process, going to inheritance of traits that quite deviates from the rule of Mendel. And this will be discussed under non-Mendelian inheritance, hence the term non-Mendelian. Now, while most pairings led to offspring with genotypic and phenotypic ratios that matches those expected from Mendel's laws, some deviations were also found. These natural deviations are examples of non-Mendelian inheritance, which could be two types. Mendel's law do not apply at all, and second, genotypic ratio is that expected from Mendel's law, but the phenotype ratio is not exactly the same or does not match with our genotypic ratio. So let's have an example. A type of non-Mendelian inheritance is incomplete dominance, as we can see. In incomplete dominance, our offspring here have an appearance that is somewhat in between the phenotypes of our two parents. So we have, our parent here is that we have two roses. One parent, the red one, carries a homozygous dominant red flower allele, while the white roses carries a homozygous recessive white flower allele. Supposedly, our expected output or expected offspring for F1 generation should also have the red coloration, because a cross between these two would be producing pure heterozygous offspring. However, in incomplete dominance, the dominant allele was not able to be fully expressed in the offspring, and as such, it displayed a different coloration. So in this case, it's color pink. We also have here codominance. In codominance, two alleles are expressed in heterozygous individuals. Co means two, and dominance, which refers to dominant alleles. So this means that two traits are expressed in a given organism. So for example, we have here two different animals or two parent animals, which the other one contains white fur and the other ones has red fur or brown fur, and their resulting offspring technically is spotted, which contains both of their colors. Another example of codominance is the flower rhododendron, wherein the flower here appears to be spotted, one that is red and the other is white. Neither of the dominant or recessive traits was able to be fully expressed, and as such, two traits were expressed in this organism. So this is also another example of codominance, wherein type A and type B are technically codominant. However, there is a recessive allele for type O, but if type A and B show regular dominance over these recessive allele, so that being said, there is a very low chances for one to have type O. Thus, the only way to be type O is to be homozygous recessive. I would like you to ponder about this question and pause the video for a while. Is vitiligo, which is the skin discoloration as you can see in this image, a case of codominance? You might want to pause the video. Alright, so actually this is not a case of codominance because vitiligo is a case of an autoimmune disease wherein the skin fails to produce pigmentation. Alright, so again, this is not a case of codominance. Our next part is multiple alleles of polygenic inheritance wherein a specific trait is controlled by more than one gene or by more than one gene. Instead, there are several genes that may contribute to the final phenotype of a given trait. So, the traits that can be passed down from the parent to offspring, there are many different variations because of that. Also, polygenic inheritance could be influenced by environmental factors. A good example of that is the hydrogenia flower, wherein normally in a normal pH it's around purple, but under acidic soil it becomes bluer or bluish, but under a basic soil it becomes more pink to red in color. Okay, now we're done with unit 2 and down to our last unit on autosomes and sex chromosomes. So autosomes are the first 22 homologous pair of human chromosomes that do not influence the sex of an individual. So I already showed you a complete karyotype of this one. So the autosomes here technically is from number 1 all the way to number 2. So autosomes are numbered and sex chromosomes are not, since sex chromosomes should be represented by either XX or XY. So an autosome is one of the numbered chromosomes, as I stated before, and in humans, they have 22 pairs of this. Now, autosomes play a role in passing down traits from one parent to another, from one parent to its offspring, because as you can tell, these autosomes may actually contain dominant or recessive traits. So if dominant allele is present on the autosomes, then the individual will express the trait. This is already discussed in the previous slide. However, sometimes the dominant allele in your autosomes may carry some variations that may lead to a disorder or to a syndrome, perhaps. Like this one, we have here progeria is an example of autosomal dominant inheritance, wherein the parents was able to pass this down from them to their children, no? So a patient with progeria have a shorter lifespan. In fact, the oldest patient with progeria was 20 years old. Huntington disease is also an example of autosomal dominant inheritance. Now, if you have a dominant traits, we also have an autosomal recessive traits in which the parents may actually pass the recessive allele into their offspring, as we can see in this one. So this one is a very good example of the autosomal recessive inheritance pattern. For example, both of the parents are carriers, but they have no conditions. They have no conditions because both of them carries the trait or carries the recessive trait. So that being said, it will not be reflected or it will not be expressed to the parents. So for example, they will have their own children someday. They will be producing offspring, and there is a slight chances that the child may inherit both the recessive traits, and at this point it will be expressed in the organism. So like all this one, albinism is an example of an autosomal recessive inheritance, albino. Sickle cell anemia is also an example of autosomal recessive inheritance. But back then in our first lecture, this can also happen because of genetic mutations, not only with inheritance. Remember that some of the amino acids during our first lecture was modified, and it also led to sickle cell anemia. Sickle cell anemia may also be a result of an autosomal recessive inheritance. Now down to the sex chromosomes. So the sex chromosomes, as discussed earlier, is actually our last pair in our human karyotype, and they are not numbered. They specifically have the X or Y chromosomes, X or Y for male, and XX for female. Now, in every animals, it's actually different. In humans, it's XX and XY, but in other animals, it varies. So the structure of the X chromosome of most organisms are straight, rod-like, and comparatively longer than our Y chromosome, as you can see in this one. This one is the Y chromosome, and this one is the X chromosome. So if we're going to compare them, the X chromosome is much larger or longer than our Y chromosomes. So the X chromosomes have a very large euchromatin and a small amount of heterochromatin. And euchromatin have a large amount of DNA. And with that being said, much genetic information are stored in the X chromosomes in comparison to your Y chromosomes. But the Y chromosome is particularly very, very important because this is where sex determination, or this is where the sex of a human will be determined. No? So in the Y chromosomes, they actually contain the PAR1 and the PA2, or the pseudo-autosomal regions. As you can see, this takes up about 5% space or region of the said chromosome in our Y chromosome. They contain genes shared with the X chromosomes. The Y chromosome also contains the male-specific regions, such as actually 95% of the chromosome contains the male-specific regions, and 5% of which contains the PAR1 and the PA2. And this one, it was stated as the male-specific regions because majority of the genes here includes SRY and the AZF genes, or the azoospermia genes. So SRY and the AZF genes are responsible for the development of testicles and necessary ducts for a male human. This one is a very good chart of how sex is determined in the case of humans. So for example, we begin with an egg and a sperm cell uniting together, forming a zygote, as you can see in that one. If the zygote inherited the Y chromosome from its parent, you can actually track it through the Punnett square. There will be a presence of the SRY gene. And because there's a presence of SRY gene, it will technically influence the development of the testes. And if the testis is developed, meaning to say there would be a presence of testosterone, Wolffian ducts, and as such, male reproductive structures will continue to develop, right, as we can see in this one. On the other hand, the presence of the SRY gene also influences the presence of MIS genes, in which its function is technically for the regression of the Mullerian ducts that soon enough that will also halt the development of female reproductive structures. Okay, if we're going to compare this one, for example, this is for female, the zygote contains the XX chromosomes, and since it does not contain the Y chromosome, it will not produce it does not have the SRY gene. So there's an absence of SRY gene, so no testis will be produced, or will be developed rather, in the process, but ovaries will develop. So if ovaries will develop in our growing embryo, for example, or growing human, there would also be absence of testosterone, there will be absence as well of Wolffian ducts, and as such, the male reproductive structures would regress. On the other hand, since there's also absence of the MIS gene, the Mullerian ducts will develop, but at the same time, because because of the absence of both of these hormones, especially this one, the female reproductive structures will continue to develop. Now let's move on to the sex-linked traits. As your X and Y chromosomes are also carriers of different traits. But, I'm sorry, your X chromosomes are technically carriers, but the Y chromosome does not carry any traits. But the Y chromosomes is very important for sex determination, as discussed in the previous slides. So many sex-linked traits are carried on the X chromosomes. No? A good example of this one is the if we're going to cross a red-eye male and white-eyed female and apply it in a Punnett square, for example, the resulting structure would be 50% red-eye female and 50% are white-eyed female. So, technically, since the Y chromosome does not carry any traits at all, whatever the alleles that are carried on the X chromosomes will be expressed in the organism. So even though our X chromosome here carries the recessive gene, since the Y chromosome does not carry any traits at all, the organism will have red eye. Or white eye rather, white eye. Hemophilia is actually another example of our sex-linked trait problems, wherein majority of the carriers are female and this is usually expressed among male patients. No, as we can see in this Punnett square, medyo malabo. I'm so sorry. In this patient, although the X chromosome only carries recessive hemophilia related traits, since the Y chromosome does not carry any traits at all, this patient would express the hemophilia, no? In comparison to this one, for example, we have two. Actually, this one would also express the hemophilia or the condition rather. But let's say that there is another X chromosome that carries a dominant trait, and the dominant trait carries a normal gene. And most likely, among female, they are less. There is a lesser chances for them to develop or to have this condition of hemophilia because they have two X chromosomes in comparison to male. Color blindness is another example of our sex-linked recessive traits. Now, lastly, let's discuss about pedigree and genetic disorders. In order for some geneticists to track if genetic diseases are inherited from one family member to another, they actually employ the pedigree charts. So this is a diagram that shows the occurrence and appearance of phenotypes. And this diagram usually uses shapes and colors to represent if a given individual inherited a given genetic disease. So we do have a rule in our pedigree charts, as you can see here. Square if it's male, circle if it's female. A horizontal line would equate that both of them are mating or both of them are parents, no? And then as we can see here, a line that is connecting our parents, a vertical line connecting the parents all the way to another shape, represents offspring, the F1 generation or children, no? So as we can see here, we also have symbols for our twins, either dizygotic or monozygotic twins. We also have here a diamond shape if the sex is unspecified, and the most important symbols or legends here are technically the male, female, and the lines, as well as if the shapes are already colored, because in pedigree charts, when the shapes are colored, it means that they inherited the genetic disease. So for example, let's make a pedigree for the following couple. Dana is colorblind. Her husband Jeff is not. They have 2 boys and 2 girls. Let's try to draw a pedigree chart on this one. So again, going back, if it's male, it's square. If it's female, it's circle. So Dana is represented by circle and Jeff is represented by square. or box, no? And then since it's explicitly stated that Jeff is Dana's husband, I'm just going to draw a horizontal line in between. And they also have 4 offsprings, 2 boys. So I'm going to illustrate 2 squares and 2 girls. So dalawang circle. Now the next question is, si Dana, she's colorblind. So I'm going to highlight Dana with black color because according to our pedigree charts, may coloration yung affected individuals. Now the real question now is, how many of their offspring or children will inherit color blindness as well. So actually you can actually predict this using a Punnett square. For a while. Oh, come on. So let's say Dana is colorblind. XX colorblind. So she's a carrier, so to speak. So for example, colorblind here, and then Jeff is not. He's normal. Let's try to have a Punnett square of that one. So I have here X chromosome containing the color blindness, sex-linked traits though, by the way. And then I have here Y, X, Y. Okay. So let's try to derive on a given answer. I have here XCX. And then XCY, XXXY. Meaning to say, 50% nung anak nila, no, 50% of their children will inherit or might inherit, especially if this one is dominant allele pa, might inherit the color blindness. And 50% naman, on the other hand, is normal. No? So going back to our pedigree charts, in order for us to represent this, our result, 1 boy will have color blindness and 1 girl. So kulayan na natin dito, 1 boy and 1 girl. So that's actually how you make pedigree charts, right? So actually that's the last of my discussion. You have assignment 2 and 3. So for these assignments, for the submission dates of these assignments, please do refer to our calendar. I know that there's too much on this lecture, so if you have any questions, please do not hesitate to ask. I would be opening my Calendly for your consultation. In the past few weeks, it's not yet open, so I'm not receiving any request from any students. But I would be opening them for if you have any questions, or do approach me right after classes. Thank you, everyone.